This is quite a substantial session, and it's divided into two halves. Both halves are to do with work that's largely been completed in a TWCF, Templeton World Charity Foundation project, which I've been involved with, which is why I'm chairing this session. And we're going to, first of all, have a contribution from somebody whose official title now is Dame Professor Ottoline Leyser, though she's rather modest and is quite happy with Ottoline usually. And she's going to be talking for about 30 to 35 minutes, and then we're going to have just 10 minutes of fairly focused questions discussion on what Ottoline has said. We'll then move straight into Michael Ruse, who's Professor Michael Ruse, who's also been heavily involved in the project. Indeed, technically, he is really one of the co-PIs in the project. And Michael will talk similarly for about half an hour, and we'll have about 10 minutes of questions. And then there's a normal break for half an hour. Then we come back. There's the third talk by Fraser Watts, who has been actually paddling incredibly hard under the water and driving it forward, the project, in many ways, and has got intellectually involved. And we'll be talking about what he's been doing, and we'll have just 5 or 10 minutes specifically for Fraser. And then there's a panel discussion, where we've got about sort of 45 minutes or so for people who want to raise general issues, to do any of the 3 presentations, or that have been inspired by them. And obviously you can ask particular questions as well. And that takes us through to lunch. So it's the whole of the war. Ottoline, I think we can probably get straight into your presentation.
Avoid embarrassingly falling over. And I will instead embarrassingly, intellectually fall over at the podium by, yes. So it's a pleasure to be here, and I've had a very interesting time so far listening to the talks and contributions and very interesting conversations as well. Okay, I will do stage voice. Good. So I was just saying how pleased I am to be here and how interesting a time I've had so far, and at the same time how hopelessly inadequate these kinds of interdisciplinary meetings always make one feel, because you are, yes, totally non-expert in the vast majority of things that are going on. And so I'm kind of prefixing my talk with the caveat that I have 0 formal training in history of science, philosophy of science, any of those things. I am here as a data point. I am someone who does science, and you can use what I say as as data for your analysis. So what kind of science do? I do plant developmental genetics, and I am therefore interested in trying to understand how single cell, in this case a plant cell, manages to become a complex multicellular organism with all the cells in the right place at the right time doing the right things. It is an extraordinary question in biology, which I find absolutely gripping. And it's also a question that is complex, and it has very many layers to that complexity, but it is also definitely tractable. I don't like it when people say, oh, it's complicated, as a kind of excuse not to think about it. The complexity is really interesting, and that's why thinking about it is so intriguing. And as I say, it is tractable. We've made huge amounts of progress over the last 200 years in understanding this kind of question. And so how have we managed to do that? That's what I hope I will say something about now. So I'm going to give a talk, and it's going to have slides, and I'm going to start with slide 1, and I am going to move forward to slide 2, and then there will be slide 3, and then slide 4, and I'm sorry it will go on longer than that, but eventually I will get to the last slide. And it will be a series of slides, and I'm hoping that through this series I will build up some kind of argument or points or series of discussions by presenting things in a sequential way. And I mention this because that's how, that is a key tool to getting into complexity, is to pick some bit start from it, move forward in a sequential way, and build on that to increase one's understanding of that complexity. But of course, the complexity has nothing to do with that linear arrangement of ideas. So that's the kind of first point, which will come back at the end, and is a sort of recurring overall theme. There are many things we do in order to get our heads round the complexity of biology that are key tools to doing that, but are fundamentally kind of contrary almost to the complexity that we're trying to study. And that is not a problem, that's fine. And that's, I think, the core message of what I want to say is you know, there's an awful lot of isms that have been discussed so far and will be discussed more. My favorite ism is pragmatism. You do what you need to do to get the job done. And one of the things you need to do is linearize a nonlinear system. So biology essentially is about how living systems work. And in order to cheer myself up in this place where I feel wholly inadequate because I don't know anything, I'm going to use examples that come directly from my own research as a kind of comfort blanket. So here is my comfort blanket. This is two plants. They're Arabidopsis plants. That's the model system that I work on. It's kind of the fruit fly or the rat for plant biology. And what you can see is that these two plants, the way Arabidopsis grows, it makes this kind of rosette of leaves, and then it sends up this elongating shoot where the flowers are at the top here and the fruits with the seed. And after it flowers, it branches, and it makes branches actually in a top-down sequence. And here's a branch that's activated and produced this elongating branch from the base of this leaf. And the way this whole process happens is that at the tip of each growing shoot, there's a group of cells called a meristem. In animal biology, they would be considered to be stem cells, which is a bit confusing because plants have stems, but this is a meristem, and this meristem is building the plant underneath it, and it makes leaves at its flanks, and within the base of these leaves, between the base of the leaves and the stem, there's a whole other meristem, and that meristem can remain dormant or it can activate. And if you do any gardening at all, you will know about this because it's why pruning your roses makes them bushy, because this main meristem is able to inhibit the activity of these secondary meristems, but if you chop the top of the plant and take away the main meristem, those secondary meristems will activate and make branches. And so the meristems essentially talk to each other and make kind of collective decisions about who should activate and who should not activate. And that's the kind of question that I'm interested in addressing. And damage by a pair of secateurs or a predator is one of the things that can come in from the environment to influence the decision about whether or not meristems activate. And the other one, which we work on a lot, that's illustrated on this slide, is essentially nutrient availability or fertilizers. So this plant here has been grown in fully fertilized conditions, whereas this plant, which is genetically identical to this plant, has been grown with limiting nitrogen availability, and it makes far fewer branches. And people tend to look at this and think this is a boring result because it is to do with the fact that there just isn't enough nitrogen to make more branches, and this is not true. What's happening is these buds here in the base of the leaves are being suppressed so that any nutrients that are available are being invested in building root, because if you're limited for nutrient availability, then the nutrients you have you put into roots because that's where you're going to find more nutrients. So these are really quite sophisticated decision-making processes that plants are having about where to invest their current resource to optimize or to improve their chances of future resource maximization. And some of the decisions going on here involve epigenetic changes, which we've heard about. But the epigenetic changes are also more generally involved in driving the developmental programs forward, and they are based on the genotype of the plant, the environment in which it's growing, and the previous developmental decisions that the plant's made. So it's a whole, there's a really rich collection of information sources that are being integrated together throughout the plant's life cycle, and we are particularly interested in understanding how that integration happens at the level of axillary meristems, so meristems sitting here in the base of each leaf, and whether or not they should grow out. And so we use things like nitrogen availability as an easy to manipulate variable that changes that decision, and we hope to understand how plants then make those decisions. Okay, so that's kind of what I'm trying to do in my research, but more generally, any complex biological question you come at in my experience, two ways that are interconnected. And the information you get from these two information sources that are interconnected are how you pick the system apart. And the first thing you have to do is to describe what you're looking at. And this you can think that you might be able to do this once at the beginning, but of course not, because your description is heavily kind of biased by what you're thinking might be going on when you start doing the description. So the parts you pick to give names even, and the names you give them are of course contingent on how you're thinking about the question at that moment. So description is absolutely essential. You have to describe what you see, and you have to give names to the phenomena, the bits and the things that they're doing, because otherwise you can't talk about it and you can't think about it, but those parts and processes that you name are inherently kind of contingent on what you happen to be thinking about at the time. So that's, I think, really important. The other really important aspect, which if there is a new biology at all, and I'm a bit skeptical about that because I think biology is about trying to understand how living systems work. It always has been and it always will be, so that's biology. The newness in the system comes from the fact that I think we've had really a very rapid acceleration in the tools available to us to describe systems. And that has allowed us to describe parts that we have not been able to describe before, and that's been very kind of revelatory. And the ability to describe or to capture or to see what's going on wraps into the whole kind of the questions that we can ask, and I'll get back to that right at the end. So I just want to emphasize now the sort of level of complexity. I keep saying complexity, but I don't like that word because it scares people. It puts people off. It gives you an excuse not to think about it, and it isn't. But what we're trying to think about is a system that is operating at a very wide range of scales. And I think this is the really interesting and exciting aspect in biology, and particularly if you're a developmental biologist and a developmental geneticist at that, this is the part that is really gripping. So just to illustrate that, this is a picture of an Arabidopsis plant living in the real world rather than in my lab, and here it is. And we've decided that this is a plant. We decided that it's one plant, despite the fact it's living in this lovely environment, interacting with all kinds of microbes in the soil and all the rest of it. But we've decided that we're going to think about it in terms of this thing called a plant. And we're going to kind of name some parts. And this is a part which I will kind of zoom in on in a minute. And it's about a centimetre, about 10 to the minus 2 metres in a kind of order of magnitude scale. So here it is. We've decided to call this part a leaf. And here it is. It's a very nice leaf. And if you zoom in a little bit more, we've decided to call these interesting kind of jigsaw puzzle-shaped things cells. And this thing, this is a rather beautiful thing, is a multicellular trichome, which is a leaf hair that Arabidopsis has. We've decided to call that a trichome. And we keep zooming in. And here's a close-up of the cell. We've decided this is a cell. We've decided these things between them that are kind of made of cellulose in plants are called cell walls. And if we move then to a shot that's based on an electron microscope freeze fracture image, we can fracture into the cell and we can see things that we've decided this is called a chloroplast. And this is actually a fractured nucleus, which is where all the lovely DNA is. So if we can look kind of into the nucleus, this is a nuclear pore that connects the nucleus to the cytoplasm, and inside is chromatin, which is this kind of folded up version of the DNA, which again we heard about from an epigenetic point of view. We're now down to 10 to the minus 8 meters, and if you keep zooming in, here is the DNA, here is the lovely double helix that we know and love. And here are the bases, the A's, G's, C's, and T's pairing across the double helix that are the genetic code, essentially. And we're now down to the nanometer scale. So we've come across 7 orders of magnitude, and that's up to a centimetre, and the plant's kind of 10 centimetres, 8 orders of magnitude. And just to get you oriented, that's the metre to the world is then fewer orders of magnitude than the orders of magnitude over which we're working. So what is really exciting is what the relationship is between those tiny nanoscale things going on in your DNA, for example, and the meter, centimeter scale things going on at organismal level. That is just a gripping question. So we can describe biological systems at all those scales quite accurately now because of the tools we have available, but a description is clearly not nearly enough to understand what's going on. So the other major approach we have beyond description is perturbation. So we need to be able to poke this system and see what happens. And so at some level, this already is a perturbation. I've got the same, potentially the same system, but I've given this one lots of nitrogen and this one less nitrogen, and as a result, changes have happened. So I can describe the differences between this plant, and that can give me some clues at what's going on. But I am fundamentally a geneticist, and my favorite perturbation is a genetic perturbation. This is, in my opinion, a super powerful perturbation because it's way less biased than all the description and all the descriptive comparisons or perturbations that I pick to put in. So what do I mean by that? So the way genetics works as a way to unpick biological systems is you take your organism, here it is, in this case, a whole tube of seed of it. You soak it in a mutagen that changes DNA bases randomly. You'd have thousands and thousands and thousands of seed that you've treated in this way. You plant them all out, and you look for ones that look different in a way that's interesting to you. So there is some bias there. You pick the ones that look interesting to you, but that bias is dependent on the question you're asking. So I am asking, how is it that plants manage to tune their branching according to the environment in which they're growing? And so the kinds of mutants that I've picked look like this. So here are 2 genetically identical plants again, and they differ genetically from these plants by 1 base pair. We spent a lot of time figuring out which base pair this is, and we know. 1 base pair, difference between this, and these guys are very, very branchy, and they're more branchy on fully fertilized conditions, and they are much more branchy on with lower N. Essentially, we have broken in this system by changing this 1 base pair the kind of resource allocation system that the plant has to decide whether or not to branch. These plants more or less branch regardless. They pretty much every single one of these buds will activate, and that's that. So they can't do this plastic response to the environment anymore because of this 1 base pair change that we've made. At this point, one could start talking about causality, but I've decided that causality is a toxic word in this conference and one shouldn't mention it. And it's also actually not very interesting in some ways, for a variety of reasons. This is where pragmatism comes into it again. I'm interested in how this works. I'm not interested in exactly what's causing what, because the whole thing is a system that is totally ridden with feedback. And so the whole notion of causality is just not worth thinking about. But nonetheless, what I can tell you is at the beginning of the lives of these 2 plants, they were seeds. And at that point, the only discernible difference was this 1 base pair. So you could say, okay, well, that's great, we've done it. We know what happens. This base pair is what you need to be able, which is clearly nonsense. And the interesting part is trying to understand why it is that changing that 1 base pair has had this big impact on the way that the plants grow and develop. What's shown here is the kind of the Watson and Crick central dogma that DNA, in this case the relevant gene that we've changed, makes RNA, and that makes protein, and the proteins do something. And I'm going to cut very long stories short from a biological point of view to make the point about how you do the science rather than what the science has found. So that the particular gene that's changed in that particular plant encodes an enzyme that does this oxidation reaction that converts this stuff to this stuff. And so the one base pair change means that this protein doesn't work anymore. This is actually a key point in genetics that is frequently missed in public discourse around genetics. People talk about, for example, the gene for haemophilia. This is not the gene for haemophilia. This is not having an active version of the gene for blood clotting. It's not that you have a gene that gives you haemophilia, it's that you don't have a gene that would prevent you from having, and that's a key difference, because the idea that there's a gene that gives you the disease is a bonkers idea. Anyway, the genetic approach essentially allows you to break the system one gene at a time. And so if you take out this protein and are unable to convert this to this, then you're branchy. So the difference between this plant and this plant is that this one makes this and this plant doesn't make this. And you can do quite a good job actually of converting these plants back to these plants by spraying them with this, which is kind of cheering, but frankly, from my point of view, still immensely boring, because it still doesn't, we haven't got beyond the nanoscale here. This is not helping me understand how how these whole plant level decisions are made by nanoscale events. And so to summarize, huge amounts of work in thinking about this. The way that we have to think about this now is as a kind of series of interlocking self-organizing systems. That's how with the current tools we have available, the current kinds of data sets that are being produced, the most useful conceptualization of what's going on for me to understand my system is a series at different scales of dynamic, self-organizing, feedback-driven mechanisms. So at a cellular level, here is this compound. It is acting through a signal transduction pathway, which is crudely represented by this series of blobs that are various proteins that do various things. And actually, essentially what this is doing is tuning the levels of another protein. And that process is regulating the allocation of a particular protein onto the membranes of cells in the plant. And this protein is actually resulting in the transport of a different hormone. This is a plant hormone, and this is a different plant hormone, that the amount of which is moving because of the presence of this protein on the membrane depends on this thing. So you can think of this cell then as this kind of dynamic self-organizing system of interacting molecules, and that system is sensitive to both local and systemic inputs and is therefore equilibrating or shifting between equilibria with particular dynamics in different circumstances. And then you can think of these meristems that I talked about. Here it is, a scanning EM of tomato meristem. It's the dome of cells at the tip of the plant making the leaves in this beautiful spiral pattern. And you can think of that as a dynamic self-organizing system of cells that is sensitive to these local and systemic inputs. And then you can think about the plant as a dynamic self-organizing system of meristems that are talking to each other. So you've got these kind of scalar stacks of regulatory networks or architectures that are operating at different scales. Those scales all talk to one another. It's not that they're separate or distinct, but it is instructive in understanding how the system works to think about those scales somewhat differently. All of our thinking is now absolutely dependent on computational mathematical models, because you can't think straightforwardly about these kinds of dynamic, self-organizing, feedback-driven systems, because we think in this linear way, both in terms, both dynamically and kind of physically linear is our favorite way of thinking. But through maths and through the conceptualization of these regulatory networks, it is possible sensibly to layer up one's understanding of the system and to focus at different levels contracting together almost, or kind of gathering together. I liked this multi-layer feeling of humanity, where the idea is to bring together things. You kind of do that a lot when you're thinking about these different levels of organization. You try to collapse them down onto one another or up onto one another, depending on the level of organization that you're thinking about. So if we go back very quickly to this diagram, I want to try to illustrate that just at this nanoscale to make some of these points a bit more loudly. So we've got DNA making RNA making protein in the kind of central dogma type way. What I'm going to try quickly to do here, is this going to work? No, it's not going to work. That's annoying. I had a movie, it's all right, I'm running out of time anyway, a movie of transcription, which we now know a huge amount about. And it's a beautiful movie that shows the enzymes that are required for transcribing DNA into RNA, both being collected at the right place in the DNA by this collection of transcription factors that comes together to mark the spot where these things have to assemble. And then it's a dynamic movie of the thing trundling along the DNA, sucking in the bases and spitting out the RNA. And it's, you know, so what I'm, as I say, I'm not really tremendously interested by this scale, but I have colleagues who've spent their entire lives working at this scale, and so this word transcription unpacks into a lower level dynamic self-organizing network of interacting things. And I just call that transcription. And that illustrates 2 points, one of which is you can work at all these different scales, and all of them are rather, all of them you can think of in terms of parts and systems. And the other is that that we've now given words, just a word, in the same way I've given a word to DNA, I've given a word to transcription. So I can also name processes, not just physical parts. And then the other thing, of course, is that we name entire sets of processes. So this is, as I say, Watson and Crick's central dogma. DNA makes RNA makes proteins. And all of this stuff has consequences for how you think about the system. So particularly as soon as you start calling something a dogma, you're in big trouble, because it's bound to be wrong. I mean, this is the take-home about all of this stuff. You have to do it. It's really useful. You cannot make progress unless you do it, but it is bound to be wrong. And so if you hardwire into your brain that this is the central dogma, when you discover that there's an enzyme which you then call reverse transcriptase, because as far as you're concerned, this is the main thing, and this backwards thing is weird and shouldn't be happening. So you call the enzyme reverse transcriptase, as though it's some kind of bizarre reversion of some, you know. Anyway, then you get very surprised and you know, well, that's okay. You can be surprised, but you have to appreciate that your notion of a dogma is bound to be wrong, and the words you've used, like transcription and therefore reverse transcription, are very much conditioned by the history of the subject, and you have to keep those well up for grabs, otherwise it can constrain your thinking, which is a very bad idea. And then, of course, the other thing I pointed out is that anyhow, this whole idea of linearity from one end to the other is completely a non-starter, because absolutely all biological systems I've ever been able to think of involve these kinds of feedbacks. And so, for example, the amount of this stuff you have actually regulates how much more you make. And this is a very classic negative feedback system for the production of things in biology, so that the thing that you make limits its own production, so you stabilize at some particular point. And then obviously the dynamics in the wiring of that system will equilibrate to a particular level of the thing, and if you poke those dynamics either environmentally or different, then you can change the level of the thing that is produced. But in both cases, that level is subject to continuous dynamic regulation. It's not that you've made an amount and there it is. You've made an amount, a certain amount of that is going away, a certain amount of that is being made, and the rate of production and the rate of degradation are resulting in some equilibration at a particular point. So this is the kind of final point, is that the place we've got to in thinking about this means that most of the interesting stuff now is happening at a level of thinking that is to do much, much more with dynamics and regulatory architectures than it is to do with parts and what those individual parts do. So as well as this kind of layering of scales, at each scale you can layer the, I mean literally from a kind of physics point of view, the order of activity. There's the thing itself, but then there is the flux of the thing, the rate at which it's being made and the rate at which it's being degraded. And there is now a kind of acceleration term in that flux is tunable by inputs and outputs that are dependent on the regulatory architecture of the system. So it's complicated, but it's tractable and is beautiful. And so getting your head around it is a joy. And the very the tools that you use are essential, and we shouldn't beat ourselves up about them, but we should always be aware that they are tools. So parts, from a point of view of parts, it is really important to remember that biological systems do not think they have parts. From a point of view of a biological system, it has no idea what a cell is, it has no idea what DNA is. It's a bunch of stuff hanging together in a very interesting non-equilibrium state. But having said that, we have to think that systems have parts, otherwise we have no hope of understanding them or talking about them to one another, which is, of course, half the fun. But the notion that you can sort of break things down into parts doesn't make sense. And anyhow, one person's part is another person's system. So because we're operating at all these kinds of levels, at any one of those levels, you can talk about parts. But you kind of amalgamate those parts together, and that thing becomes a part at the next level. Then, of course, to understand how this thing works, really the whole thing's about dynamics, as I described, and the interactions between the parts dynamically, and also architecturally, how those interactions play out. And you can label those interactions and increasingly the dynamics also as a kind of part. So one of the interesting things happening in biology now is the labeling of regulatory architectures so for example, different kinds of switches. You can have many different ways to switch from one thing to another way. You can go very gradually, in a way that you could easily go back. You can go very suddenly. You can be thresholded. And there's a whole range. And now we're beginning to invent words to describe those kinds of switching mechanisms, which are then those kind of those third-order kind of cubic descriptions, I suppose. So we've got multi-order descriptions, or multi-order. You can think of parts both in terms of the scales of physical parts, but also in the terms of orders of dynamics. Okay, and then of course there's all these lovely nonlinear feedbacks, whereas we like to think in this linear way. And you'll be pleased to know I'm now coming to the end of the talk, you see. And hopefully the fact that I've laid it out in a vague linear way that builds will have made it easier to get one's head around than if I'd just sort of randomly shown the slides in some order or other. So to really understand biology, then you need this kind of integrative thinking. You have to, on the one hand, you have to go with the way you can think most easily, linear interactions between parts, and that's super useful, but only if you know all the time that it's wrong. That's the key to kind of keep in your head. It will definitely not be right, but it will definitely be useful, and that's okay. And then all of this has been going on forever. New technologies, I really liked Donovan's thing at the beginning about how we can see more deeply. And I think that's what's new in biology, is the new technologies that are allowing us to see more deeply and therefore move from these into this cubic level of interaction, thinking about the properties of the switching or the properties of the transitions or even the properties of the stable states dynamically rather than as the individual bits. And so when I was putting this together, it occurred to me that if we were going to do reductionism in biology, I don't think thinking about parts is reductionism, because as I've said, the whole point is to use that as a tool to understand how the system works. The reductionism in biology, the important part is to pick a tractable question. You can't possibly address the thing head on, here we go. You've got to pick a bit that you know you can make progress with. And you hope you pick a bit that's a really good example that will be rollable, outable, and you pick a bit where the tools you have available to you at the time, you know you will be able to use to make progress. So that, I think, is the key to reductionism in biology, is picking a tractable question, and if there's a new biology, it's that the questions that are now tractable are more interesting than they were a few years ago, which of course is always the case, but maybe the rate of acceleration now is higher. Thank you. So I'm doing some questions now, I remember.
We do. I was desperately trying to type down that last sentence, which I rather liked as a summary. We've got now just sort of up to 5-10 minutes questions if we want to before Michael Ruse speaks. Paul is very kindly hovering with a microphone, so in the usual way, would you just wave a hand so many of us can see it? Yes, Paul.
I thought this was absolutely brilliant.
Can you hear it back or not? Either much louder or here's Paul's microphone. Thanks.
Thank you. I thought it was absolutely brilliant.
Thank you.
Could you tell us what could be new with the new biology then?
What could be new?
Yeah.
Like I say, to me, probably my favorite new bit is the need to invent new words, new parts that describe dynamic behaviors, that clump dynamic behaviors. There's an extent to which something like transcription is that sort of word, but it actually isn't, because the word transcription really means the process by which you copy DNA into RNA. I'm interested in terminology that is to do with how the switches is perhaps the clearest example that I mentioned. The different sorts of switch you can have. Before we'd have said there's a switch. This gene is switched on, for example, in this kind of circumstances. Now, what I hope we are moving towards saying is that this switch is switched on according to this set of dynamics, and we're beginning to kind of label those dynamics. At the moment, there's a lot of borrowing of terms from engineering to describe regulatory architecture, so people are beginning to talk about coherent and incoherent feed-forward loops and things of this sort. So I was very interested in this talk yesterday that was discussing the kind of amalgamation, or the way that engineering and biological sciences are interacting, and it's useful, but I think we're going to have to change those words, because I don't think they have enough richness actually to deal with the things that we're now needing to describe, and so that, I think, to me, that's the exciting new bit, is moving up the kind of physical, the moving from static to moving to accelerating kind of parts, I guess.
Thanks. I had a quick comment about the naming thing, and then I have a question. So the comment was that, so I wonder if you would consider, borrowing some humanist colleagues to help you with these naming processes. Because I remember when I was doing my retraining, because I was a humanist theorist, and then I did some training in biology, some of the terms or some of the words they had sort of used to name some of the genes and the proteins, it sounded like it was a bunch of TWIC graduate students laughing over beers. And so I just wonder then if it's worthwhile finding people who actually are trained in language and the production of meanings to help you come up with terms. So that would be my first comment. And then the other one was, I have a colleague at UCLA who does sort of science studies, and she works a lot with geneticists, and so her name is Hannah Landecker, and she's developing an argument that's quite akin to yours, and it's very interesting because she wants to talk about the genome as metabolic processes rather than things, and I was wondering about your thoughts on thinking about them as metabolic processes given your attachment to the processes here.
Okay, so I can talk about those two things. So the naming of things within biology is already, I mean, has always been contentious. And the bonkers naming of genes actually has a very good reason for it. And I'm a geneticist, so I'm quite keen on the bonkers naming of genes. So many genes that were discovered through genetic screens are named after the phenotype, what the thing you selected was. So people like to name their genes kind of interesting things for that reason. So there's lots of examples of that. So in Drosophila, for example, there's a mutant that's hypersensitive to alcohol. It can't break alcohol down very easily, so it essentially gets drunk very quickly, and that gene is called cheap date. So there you go. So you're right. It's graduate students. They have to have some fun, for God's sake. So why not? But it becomes a problem if those genes turn out to be clinically important, and then you've got doctors earnestly explaining to their patients that they are having some problem with their cheap date gene, and this is not really a good place to be. But I think what that illustrates is that it depends very much. I mean, there's no such thing as a universally good name for precisely the reason that I've talked about. The naming of genes after mutants is actually very helpful for, in my opinion, for scientists, because it tells you immediately what happens if you don't have that gene, and in a kind of rich way. The biochemists, bless them, don't like those names because they like the biochemical activity of those genes, so they would like to call that gene alcohol dehydrogenase, which, whilst it's much more straightforwardly descriptive, at the level, that nano level, tells you nothing about the macro level. So it depends what level you like, what name you like. And so I think it's an interesting idea to involve other people in deciding what to call something, but at the same time, I think it's very unlikely that one will be able to come up with a name that will not be contentious or will be equally useful to everybody. So it's difficult. Plus, there's the speed thing. I mean, you're discovering these things really fast, and if you had to hold a conference every time you wanted to name something, it would be a bit of a problem. So actually what happens is you hold a conference in about 5 years' time when the nomenclature has become so messy that nobody understands it anymore, and you rename everything and everybody's happy again. But which does happen, particularly, for example, if you come across the same gene through several different mechanisms, and so then the same gene has several different names. Okay, and then the second question you asked me was about whether to think of genomes as a kind of metabolic state. And at some level, the answer to that is yes. So I think the kind of general conceptualization of genomes as really stable things has been dead for a very long time. Not so much actually. I mean, the kind of epigenetic changes that you were discussing in terms of the ones that are influenced by the environment in specific ways are actually a very small proportion of the epigenetic changes. A lot of them are to do with keeping transposable elements off. So most genomes of organisms that have been around for a long time and are relatively slow in development, so these have accumulated over evolution thousands of these transposable elements which hop about the genome, which means that, I do a lot of public understanding, I don't like public understanding, public engagement on GM. And one of the things you can say to people is it's very likely that every cornflake you ate for breakfast this morning is genetically different from every other cornflake due to the insertion of a bit of DNA in an unknown place, because there are thousands of these transposable elements in the maize genome, and they will move around in unknown ways. And even though they move very, very infrequently, because there are thousands of them, they will move. And the thing that keeps them moving very, very infrequently is epigenetic changes. And there's at least one theory that the whole of the epigenetic system is actually a kind of genome defense system that is there to, So I mention that because I think the problem is the concept of the genome as a super stable kind of thing. The metaphors of books and you know, those metaphors are not good because it makes the book look like a permanent thing. It's more like a Microsoft Word file that's corrupted.
Well, there's only going to be time now for one final comment or question, and that's this person here, yes, right at the other side of the room. But to emphasise, we will have this proper panel. There'll be 40-45 minutes after the third talk.
Thank you very much for a very enlightening talk. I was intrigued by your comment, causality is not really worth thinking about. I understand that to mean it sets us onto the track of linear causality, which may be a problem if you're trying to figure out what is it in the plant that makes it grow stems in other places, etc., because it's probably not just one thing, it's a bit more complicated process. But on the other hand, if you give up on the term causality, that concept of linear causality might linger on and it might come back with a revenge. So would it not be more helpful to reform that term and make sure it's understood in terms of, in a systemic way, as something like circular causality, perhaps?
Well, so my kind of irresponsible graduate student answer to that question would be, well, you guys figure out what it is, come back and tell us, and then we'll use it in the correct way. So I mean, there are so in this whole genetic approach is quite interesting from this point of view, because what you're doing is breaking one part in a feedback regulated system, and there's a famous case in plant biology now of a very elegant and beautiful set of interactions put together based on what happens if you take out a whole set of genes that is the mechanism of the biological clock, the circadian clock in Arabidopsis. And it went forward for years on this beautiful system of things positively regulating one another in this series of 3 interacting loops that was built on what happened when you broke these bits and where the system reconfigured under those circumstances. And eventually, it transpired that the entire thing was backwards. All the interactions that people thought were positive were negative. But if you have a whole feedback loop of positive interactions and a whole feedback loop with kind of negative interactions, it comes out, you can't, it's very difficult to tell the difference between those two things with stable perturbations. You have to have time-resolved perturbations that you can do sensibly before you can see it. And this, this becomes an interesting question in the context of causality. And so, you know, okay, I'm interested in how the system works, and I don't really mind what I call it, as long as I have words that I can describe it that other people can understand. And it seems to me the problem with causality at the moment is that it has too many meanings, and they're too different for too many different people. So when you use it, you confuse people rather than help people.
Can I say thank you so much? You managed to start by teaching us an awful lot of really good, interesting biology, and despite your modesty saying you won't give anything about linguistics or history of philosophy, we seem to have got into quite an appropriate set of discussions about that. So thank you ever so much. Almost no break. Michael, when you're ready, if you'd like to come up, please. Oh, so that was not a wise thing to have done. Do you know what I should do now? This is, of course, intended. This is to model some of the points that Ottoline was talking about, the problems with linear behaviour in her talk.
So let's get rid of mine. Let's get
Really
Michael's
sorry.
back up. That's okay.
Oh, thank you.
Thank you.
Good.
Well, thank you. Those of you who are film buffs will recognize the echo in my title. It is, of course, a riff on a great pre-war cowboy movie starring Marlene Dietrich and Jimmy Stewart, and I'd like to dedicate this talk to their memories. What I'm going to talk about this morning is 2 root metaphors. I'm not going. What I'm saying, sort of basically, is nothing new. I don't think any informed historian of science would differ at all from anything I'm about to say. Perhaps I've got a little twist at the end, or I'm a philosopher. I'm interested in what it all adds up to. But as I say, I don't look upon what I'm talking about as in any way controversial at all. There are, in the history of Western science, two basic metaphors, root metaphors, ways of looking at things. One is the idea of the world as a machine, mechanism. And the favorite example, one which is used by Robert Boyle, is of a clock, of the world as a clock. But it's way older. The Stoics were using it, for instance, so it's way older than Boyle. What I'm showing you is the clock in Strasbourg. What do they call it? The greatest German cathedral in France. And it is, in fact, referenced by Boyle. It goes back, I think, to the 14th century, but this particular clock was built in 1574. And it's one of those clocks like, if you've been down to Wells, you'll see it, everything goes round, and on the hour, little knights come out and get knocked down and all of that sort of thing. So the one metaphor is of the world as a machine. The other metaphor is of the world as an organism, the organic metaphor. And that's what I mean by organicism. Organicism is the idea of the world as an organism as opposed to the world as a machine. And I think it's fair to say, I think maybe my chair might feel a little uncomfortable with this, but since we're at a meeting of science and religion, it's probably appropriate to do it here. I think it's fair to say that basically Christians, certainly Christians of my acquaintance, feel at some level that there's almost an ontological or at least a theological priority to the organic metaphor. It doesn't mean to say they never do mechanistic work, but that somehow there's something about the organic metaphor which is, as I say, more Christian friendly. That it, at some level, it's warmer, it's life-giving, it's more natural, it's linked to growth. It's one, you'll see the third point I've got down, I'll talk a bit more about this later. It respects nature in some way. If we're going to deal with issues like global warming, then the organic metaphor is a better one to use. And also, the mechanistic metaphor seems to cut off free will, possibilities of choice, because everything is just going around like a clock. I mean, the clock doesn't have any choice about whether or not it's 10 o'clock now. The clock can't get up and say, no, I think I'll make it 12 or something like that. The clock's got no choice. And it seems that the mechanistic metaphor, or the world as a machine, particularly as applied to humans, excludes free will and things like that. And of course, that destroys our relationship with God and things of this nature. So as I say, my sort of background presupposition is that at some level, I think a lot of Christians argue that the organic metaphor is a more comfortable one. And since we're sponsored by the Ian Ramsey Centre, I think that this would be very much the position of the late Arthur Peacock, for example. I argued this one many times with him. Now, the organic metaphor is the earlier metaphor, and it's one, as I say again, I'm not saying anything that historians of science would dispute. They'd insist on it very strongly. The organic metaphor was certainly the dominant one in Greece, and it was particularly the dominant one of the great philosophers Plato and Aristotle. Plato, for instance, throughout his philosophy, is using the idea of organisms, organicism at some sort of level, and seeing everything flowing at some level from the one in some sort of holistic sort of way, that everything is a part of everything. And particularly in the Republic, I'm showing you a fragment of the earliest fragment that we've got of the Republic, that everything is integrated through the idea of the form of the good, the one. This, of course, is the form, the idea that St. Augustine takes up and identifies with the Christian Godhead. But particularly, for instance, in Plato's Timaeus, that this is all about the world as an organism. The Timaeus, incidentally, is an interesting dialogue. I mean, today, I think that Plato's scholars would say it's one of several. It's not the dominant one. I mean, I think that they would say the Phaedo and the Republic and probably others are at least equal if you're going to talk in that kind of language. But the interesting thing about the Timaeus was that it was the only dialogue that was known in the West until the Middle Ages. And then they went back to the libraries and discovered all the others. But it was the Timaeus that Plato was known by. Because people knew of Plato's ideas, but mainly through the Neoplatonists like Plotinus. For instance, Augustine, the great philosopher of Platonism, didn't read Plato. Apart from anything else, he didn't read Greek. So he got all of his ideas, say, through Plotinus. So the one that was known directly by Plato was the Timaeus, the idea of the world as an organism. And it's the organic model, of course, also, which is so terribly important to Aristotle. No great surprise there, because Aristotle was, before he became a philosopher, a practicing biologist. And I think that today's historians of Aristotle would say he was a very good practicing biologist. And certainly he made much of the idea of the I and of final causes and the importance of understanding in terms of ends in an organic way. Interestingly, Aristotle did not subscribe, however, to the idea of the world as an organism. He did not go along with that because at some level this would give the world some kind of ontological status that he wanted to give only to the unmoved mover. And the important thing about the unmoved mover is that it's perfect, and so it only contemplates the perfect, which means it only contemplates itself. So, in fact, the unmoved mover would be quite probably not only indifferent, but would not know anything about the world as such, and would not know anything about us. Whereas, of course, for Plato, the whole point about the world as an organism is that it does have concern for its parts and does have concern, amongst other things, for us. So, as I say, although Aristotle is deeply into the organic metaphor, he doesn't have it in quite the same sort of way that Plato has. Well, of course, to continue the cowboy metaphor, the shootout at the OK Corral came in the 16th century with the work of Copernicus, and particularly Copernicus and others around him, like Kepler and then Galileo, where in came the idea of thinking of the world as a machine, the mechanistic viewpoint, and out went the whole picture of the world as an organism, and out went final causes. And of course, people like, particularly, I think the greatest philosopher of the scientific revolution, René Descartes, was very contemptuous of the whole idea of trying to explain things through ends. And his metaphysics, of course, was dualistic. He talked about res extensa, meaning the world, the physical world, and then res cogitans, meaning the world of the mind. And res extensa, which is the physical world, the world that the scientist understands, has no mind, has no final causes, is just, if I say dead matter, that's not a good metaphor, because that would assume that there could possibly be live matter. It's just is. And that's what the world is all about, the world as a machine. However, and of course, this was given its scientific expression most fully in the work of Isaac Newton in the second half of the 17th century. However, I won't say more subtle thinkers, but certainly those who were thinking about things in a broader perspective, and particularly Robert Boyle, who was a deeply committed mechanist, recognized nevertheless that that could not be the whole story. That when one came to organisms, people like Boyle and others, of course, saw that there were problems here, that it was going to be very difficult not to think of organisms holistically, not to think of organisms in terms of, as I say, organically, in terms of final causes, in terms of what in the 18th century was called teleology. Boyle's solution to this was to recognize this, but his solution, not entirely happy one, was to say that if we're looking at the eye from the point of view of science, then we must look upon it mechanically. We must ask only causes that brought it about. That's the level at which, so we can look at the eye from the point of view of physiology and things of this nature. If we look at the eye, and we must, from the point of view of organicism, from the point of view of ends, in other words, what is the eye for? We are no longer doing science, we are now doing theology. So in other words, Boyle's solution was, I don't think it was necessarily only Boyle's, but the solution was to say science deals with the world as a machine. Inasmuch as we're thinking of the world at an organic level, and Boyle didn't say that we can't. In fact, Boyle said we must, so he wasn't denying this. But what he said is we're no longer doing science now, we're doing natural theology. And those of you who know your British history will know that this was a very happy solution because natural theology was a very important part of English theology as the middle way, the via media, between the authority of Rome on the one hand and the sola scriptura particularly of the Calvinists on the other hand. And so, as it were, the middle road that they wanted to take through this was the British way, and this was one which made a great deal of empiricism. And so, inasmuch as you are looking at the physical world, you are doing science, but you are also doing God's work, because you are also doing natural theology at the same time. And this was a continuing problem. Mechanism seemed to work just tremendously right through the 18th century, one triumph after another. But still, there was the problem of organisms, and this was one that probably the greatest philosopher of modern times, Immanuel Kant, wrestled with. And in fact, I'm not sure that Kant ever solved it properly. If you read, particularly if you read the first critique, there Kant is arguing that the laws of nature, the laws of Newton, are in some sense necessary, and he's got a perfect picture. Then, of course, in the second critique, he turns to morality, the critique of practical reason. But still there's the problem of organisms, which he turns to in the second half of the third critique, the critique of judgment. And Kant recognizes that this is a problem, and basically he doesn't have a solution. Or rather, he does have a solution to say, well, of course you can do biology, but you're not doing quality science. And Kant's famous statement was, there will never be a Newton of the blade of grass. He recognized that the biology was there, but his solution, because Kant, you see, didn't want to bring natural theology into his science, or even give natural theology a particular status. Because Kant, I think, represents the more radical side of the Reformation. He was a pietist, and for him, as later people like Kierkegaard and others like that, he did not want to get into natural theology because that at some level would destroy faith. Kierkegaard's quite clear about this. If you can prove God, who the hell needs faith? Faith requires this leap into the absurd. And of course, Kant is writing before Kierkegaard, but I think he's sharing this idea that at some level, we've got, what does Kant say? I had to destroy reason in order to make room for faith. And so that means that for Kant, natural theology, as he makes very clear in the first critique, is an absolute no-no. And then he gets to the third critique where he's dealing with organisms, and he can't take the Boyle strategy out of saying, oh, well, it's okay, folks, we do science on the one hand and we do natural theology on the other. Kant's happy to do the science, but he's not happy to do the natural theology. So he's caught, and the only solution he's got is at some level to downgrade biology and to say, well, biology, we do it, but it's not real science, not good science. I must confess, the kind of attitudes I was exposed to in England in the 1950s, when I was growing up, I have to tell you that the bright kids did maths, physics, and chemistry, and I'm afraid that biology was for what they described as late developers. So this is the unhappy situation as we go into the 19th century. Of course, there were those who were perfectly happy with this, particularly the British natural theologians, people like Archdeacon Paley, who made a great deal of the fact that the only way to understand things like the eye is in terms of God. So Paley and others were quite happy to do this. And then, of course, along came Charles Darwin, who was determined, and I think quite literally, I think Charles Darwin was quite determined to be the Newton of the blade of grass. I mean, one of the things you have to understand is Darwin was an awfully nice, friendly sort of chap, but he was driven. You know, we've all had graduate students who, you know, just absolutely driven. And Darwin was driven from day one, as his mentors recognized. And of course, Darwin not only came up with evolution, but he came up with the mechanism for evolution, which he expresses in his great book on the origin of species, namely natural selection. And what Darwin did was give a mechanistic explanation of design. In other words, what Darwin set out to do quite explicitly and felt that he had done was to bring organisms beneath the machine metaphor. So that's what he set out to do. And of course, that's what eventually other scientists, other biologists accepted. And of course, we've seen one triumph after another until, of course, it ended with molecular biology, which is all very much a continuation of this. And so I think it's fair to say that today's biology is, in many very deep respects, as we heard from the first talk this morning, deeply mechanistic, deeply within the machine mechanism. And if I can introduce Richard Dawkins to this particular group. I mean, one thing about Dawkins is that he's very good at expressing sort of basic positions. I mean, give him great credit for being really quite brilliant at this. And particularly Dawkins recognizes this. And of course, he wrote a book called The Blind Watchmaker. In other words, how biology now, how Darwin, thanks to Darwin, we can now explain organisms in terms of watches, in terms of clocks. And if you go back to the selfish gene, he says this quite explicitly. We are survival machines, but we does not just mean people. It embraces all animals, plants, bacteria, and viruses. We are all survival machines, but for the same kind of replicator. And notice here comes the molecular biology. Molecules called DNA. There are many different ways of making a living in the world, and the replicators have built a vast range of machines to exploit them. A monkey is a machine which preserves genes up trees. A fish is a machine which preserves genes in the water. There is even a small worm which preserves genes in German beer mats. DNA works in mysterious ways. I love your fact. You know, if you read Dawkins, the Christian resonances come through again and again and again. God works in his mysterious ways. It's one to perform. His rhetoric, well, more recently, if you get to the God delusion, the rhetoric is out of the Old Testament. But I swear the rhetoric of the selfish gene is out of the New Testament. So this then is the situation that we've got. And seemingly, we've got a total triumph of the machine metaphor over the organic metaphor. And all those nice, warm-hearted Christians who want something else, well, you know, it's like Christianity itself. It's, you know, it's a dying thing. It's on the way out. And we understand them, we appreciate them, but frankly, folks, they're wrong. Well, of course, not everybody agrees with this, and not everybody agreed with this right back to the time of the scientific revolution. Carolyn Merchant, whose book expresses this in some detail. She writes, the female Earth was sent... What she's talking about is the situation before... Because her book is called The Death of Nature, and her book is about the triumph of the machine metaphor over the organic metaphor. And what she's doing is expressing the situation before the machine metaphor triumphed, as it were. The female Earth was central to organic cosmology that was undermined by the scientific revolution and the rise of a market-oriented culture. The 16th century, for 16th century Europeans, the root metaphor binding together the self, society, and the cosmos was that of an organism. Organismic theory emphasized interdependence among the parts of the human body, subordination of individual to communal purposes in family, community, and state, and vital life permeate the cosmos to the lowest stone. So what she's saying is that's the picture we had, and that's the picture which has gone. But of course it didn't go. In fact, one can trace, and again, as I say, I'm not I'm not doing history that nobody else has done, so I'm following in the path of historians of science. In fact, it didn't go. I won't talk particularly about Kepler here, but if you look at Kepler, Kepler's most interesting. Kepler's completely committed. Here's one of the heroes of the scientific revolution, one of the heroes of the machine model, and rightly so. And yet, at the same time, he was completely committed to the organic metaphor. If you go into the 17th century, there are, of course, the Cambridge Platonists, who go right back to Plato. You see, this is the time when people now had learnt to read Greek and could go back and read the originals, which, of course, they did. And they read Plato. And they read the Timaeus and other works like that. And so you find people like Henry Moore and others like that who are deeply committed to the organic metaphor. One of the things, I'll just add this as a sort of a side tantalizer, is the relationship between Henry Moore and his best friend at the University of Cambridge, Isaac Newton. And there's considerable suggestion that, in fact, Newton himself, the hero of the machine picture, was in fact, in respects, deeply influenced by the organic metaphor, particularly in his notion of action at a distance, which of course the Cartesians, as true mechanists, loathed and detested. And the only way that Newton and the Newtonians could fight back was not on ontological grounds, but was on pragmatic grounds, instrumentalist grounds. They said, oh yes, but we can make predictions a hell of a lot better than you can, so go with our theory. And people said, you're right, and they did. But notice that at the heart of Newtonian theory is this rather mysterious notion of action at a distance, which people pointed out, isn't really mechanism. It really, the Cartesians said right down the line, this isn't playing fair if you believe in machine models. So as I say, even there, it seems that there's a good case there's at least a case to be made for saying that the organic metaphor was not quite as dead as many thought. Come to the German Romantics, and of course, they just absolutely pushed organicism. Absolutely, people like Goethe absolutely pushed organicism over mechanism. And I think the key figure is not Goethe, I think the key figure is Friedrich Schelling. Again, I'm not alone in saying this. This is not Michael Ruse's own personal opinion. And Schelling's a very interesting character, starting with the fact that at the age of 16, he wrote a 60-page essay on the Timaeus. At the age of 16, he wrote a 60-page essay on the Timaeus, the key work in organicism. And of course, Schelling all his life pushed the organic metaphor, pushed the idea of organicism through and through. What came to be known as hylozoism. The world is an organism with a kind of world soul. It's a term, as I say, which is used, goes back to one of the Cambridge Platonists. I don't know how common it's used today, but certainly Schelling was a key figure. One of the people who, interestingly, I think was most influenced by Schelling was none other than Rudolf Steiner, the founder of the Waldorf school system, and also the founder of so-called anthroposophy. And Steiner worked in the Goethe archives, and my family's all into Steiner, so I know about this. And they all talk about Steiner's debt to Goethe, and of course it's there. But in many respects, I think Steiner owes a greater debt to Schelling, who I should say that he recognized quite explicitly as an important influence. And certainly if Steiner, for instance, when he gets into his biodynamic agriculture in the 1920s, makes explicit reference to Schelling, and particularly quite openly identifies the world as an organism. So you've got that whole tradition going that way. And I mentioned Steiner particularly because I'm going to bring it up again in a moment. If you go to America, this organicism is particularly important. Peirce, of course, is one of the founders of pragmatism. But in fact, any scholar of Peirce will say Peirce is a very strange founder of pragmatism, because apart from everything else, although he was an evolutionist, he did not like Darwinian evolutionary theory. He thought it was the greed philosophy. He didn't like Darwinism at all. He was much more into that kind of Germanic, romantic, unfurling notion of evolution, which you find in people like Haeckel and others like that. And certainly, Peirce himself says, I was born at the time when Emerson and friends were disseminating the ideas they caught from Schelling and Schelling from Plotinus. So as I say, I do think if you look at Peirce's work, you'll see a great deal of organicism there. Much more so than I think than in William James, for instance, or in Dewey. I think I think James is a much better Darwinian. I mean, I think James is much more on that side of the thing, but certainly Peirce. And of course, people like Thoreau, and of course, this is particularly in that very influential American environmentalism, probably Aldo Leopold, one of the great heroes in environmentalism, is quite explicit about his organicism. This is an unpublished essay he wrote in the 1920s. Philosophy then suggests one reason why we cannot destroy the earth with moral impunity, namely that the dead earth is an organism possessing a certain kind of degree of life which we intuitively respect as such. So, as I say, organicism is very much part of American environmentalism. I'm a member of the Sierra Club, and I'm quite sure that 95% of my fellow members are totally and utterly committed to the idea of the earth as an organism. I'm quite sure of this. The particularly interesting case is Rachel Carson, who, of course, was the most influential and important environmentalist in American history in the 20th century. And Carson, if you read, Carson was a very skilled science writer. When she wrote her great work, she'd already written a number of popular science books, and she knew that when she put forward the idea that DDT was a killer, Silent Spring, when she put forward this idea, she was going to run into huge amounts of problems with chemical manufacturers and others like that, which she did. They just descended on her. So she knew she'd got to be very careful about presenting her theory. It's pretty clear that a major influence on her was a woman called Marjorie Spock. They all belonged to what we would now say was a woman's lesbian network. I mean, they didn't use those terms in those days, but there was a close bonding of women, and Marjorie Spock and Rachel Carson were in this. And there's a huge correspondence between Marjorie Spock and Rachel Carson. Marjorie Spock is the younger sister of Benjamin Spock, the pediatrician. But more interestingly for our purposes, she was an avid follower of Rudolf Steiner. She'd gone to Dornach in the 1920s and learnt eurythmy, that rather strange modern dance thing that the Steiner people are into. And that's what she taught. And she fed to Rachel Carson huge amounts of material about DDT. That they were onto DDT in the 1940s, the Steiner people. And she fed a huge, and Rachel Carson acknowledged it, but in her introduction, there is no mention of Marjorie Spock. And they knew why that was. Marjorie Spock didn't take it personally. They knew that if she mentioned that she got it from Steiner, she was in big trouble. And so that's all omitted, but it's pretty clear that Silent Spring owes a huge amount to this, which, as I say, ultimately goes back to Schelling and back to the Greeks. Another, I think, is also, I don't think that Ed Wilson is a follower of Rudolf Steiner. I don't think Ed Wilson's read any philosophy at all in his whole life. At least if he has, he's got it all wrong. But certainly, this organicism metaphor is very strong in Ed's thinking. I don't know that he's an out-and-out supporter of the Gaia hypothesis, but he's certainly very close to it in many respects. Of course, talking of the Gaia hypothesis, the person who is the pusher of the Gaia hypothesis of the old Earth as an organism is James Lovelock. And once again, very interesting connections there. Lovelock became convinced of the world as an organism in the 1960s, from looking at planet Earth as opposed to planet Mars and seeing that the Earth was in homeostasis. But why did he persist in this? He was not an academic. He was living in a small village working on his own. He was a great inventor. And the way he made his money was that firms like Pi employed him to go and talk to them 2 weeks every year or 2 weeks every quarter. And they'd show him problems and he'd give them suggestions. And it was worth their time to give him £6,000 or £7,000, which was quite a lot of money in those days, to keep him on their books. And I mean, it was a proper arrangement. It was a good arrangement. Lovelock's a genius. I mean, there's no question about that. But who was the person he was talking to in his village all the time? It was none other than William Golding, the author of Lord of the Rings. Golding also was a bit of a loner. What? Lord of the Flies, yes. I mix up these Christian books. Thank you for that. Thank you. Thank you, Headmaster, for that correction. Lord of the Flies. And Golding was no longer an anthroposophist by that time, but in the 1930s he had been. And if you look at some of his books, particularly The Inheritors and Pincher Martin, there are huge Steiner themes in that. And we know that he was always sympathetic to Steiner. And I'm pretty certain that he passed these views on. I'm only going to be a couple more minutes, okay? I'm pretty certain he passed these views on to Lovelock. I interviewed Lovelock, and of course he denied it. But at the same time, he knew a lot about Steiner, because apart from anything else, he and his first wife sent one of their children to a Steiner school. The kid was handicapped, and Steiner schools do run handicapped schools, and he spoke very warmly of it. So I know that Lovelock knew a great deal about Steiner, and I'm pretty certain, if not directly, certainly from Golding, his confidence in the Gaia hypothesis was certainly reinforced by his friendship with Golding, no question about that. And of course, it's also been taken up by some of my favorite characters over on Zell Ravenheart, a wizard out in California. I also interviewed him. I'm glad to say we did not have to go sky-clad, that's to say without any clothes. I'm a little sorry that once in my career I did not have to take my clothes off in the cause of scholarship. I've taken them off for other reasons, but never in the cause of scholarship. I'm a little, seemed to be a missed opportunity, but they too, of course, are deeply organic, or into organicism and that sort of thing. So, what I want to say, and as I say, I draw to a conclusion now, is I want to say that although it's been a minority position, organicism has been a very hardy plant, if you like, something of a weed that people couldn't get rid of, choose your particular metaphor like that. So I think I want to end up with, particularly in the same position as the first speaker, at some level. I want to speak up for mechanism and organicism. I don't think it's an either-or. I don't think it's an either-or. I don't have time here, but I think it plays out in the ongoing debate that biologists have between form and function, holistic versus breaking it down and looking at the particular parts and how they work. So I think, for instance, the Darwinian explanation of the beak of the finch is something which is deeply reductionistic, which is mechanistic, which breaks it down, which sees that the finch has a big beak or a small beak, the kind of work that's been done on this, because of prior causes, it's all mechanical. But I think also that when we look at bigger questions or other questions like global warming, then what we need is not a position which is anti-mechanist. I don't see it as that, but which at some level has to be holistic. And just this morning, I had a thought, and I just Googled through the pictures on global warming. I got one picture after another, or one diagram after another, which is showing that you can't just break it down. You've also got to build it up as well. And in order to get the full picture, that what you've got to do is see the parts, but parts of a bigger functioning whole at some sort of level. So that basically is where I'm going out at the end. And I'd like to think that, basically I'm going out where I came in, namely with you. Namely, I want to say that don't knock the world as a machine, but at the same time, don't knock the world as an organism. but see if the two can't be brought together in some sort of holistic sort of, or some sort of holistic sort of way. And since I went out, came in with the movies, let me go out with the movies. Thank you very much.
Tremendous, thank you. So one or two questions, comments, thoughts to Michael before we officially break. Paul, is somebody just quite close to you?
Thanks a lot for a wonderful talk. Sorry if I may just answer a very brief comment. Kant did not say that he had to destroy reason or knowledge. He said he had to use the German word aufheben, which is rather a different thing from destroying.
It's a terrible mistake to say anything in front of Germans about Immanuel Kant, because immediately you show me that I'm wrong. I'm wrong, I apologize. But the point he was making, surely, is that we've got to get rid of that kind of enterprise that was going on. I mean, he goes after the ontological argument, he goes after the teleological argument, he goes after the cosmological argument. He wants to get rid of all of those. And of course, what he says is we get to God through the moral argument. The only way that we can get morality is by presuming a God. But the point that Kant wants to make is he wants to keep his science and his religion separate in a way that I don't think people were doing up to that point. And incidentally, I'd say that this is a picture, an idea, a thought that is picked up very strongly by the English historian and philosopher of science, William Whewell, who always insisted on this. And of course, Whewell was a great mentor of Darwin. So I don't think Darwin read the Critique of Pure Reason. I know he didn't. I think he read the Metaphysics of Morals, but I know he didn't read the Critique of Pure Reason. But he was always deeply committed to that Kantian point that we've got to keep our science and our religion separate.
Anybody else? Looks like it's also quite close to you, Paul. Just a bit back.
Ah, my Welsh friend.
Thanks for the talk, Michael. I just wanted to suggest that one area of research and science at the moment that really fits in with your argument in terms of connecting the mechanistic and the organicist is psychedelic science, which also fits in with the general theme of science and religion. You have mechanistic psychedelic researchers who look at psychedelics very much at the kind of molecular level. What happens if we change this molecule, that molecule? And then what happens when you take these different molecules, you get this much more kind of grand, encompassing, holistic vision. So I just think it fits quite interestingly with your approach.
Yeah, thank you. Yes. I agree. There's a man down here.
And then, thank you. Then we'll have a final contribution now. As I keep on saying, come back after the coffee break.
Well, yeah, in some ways, my thought was leading from that. Do you think there are, can you say something more about the kind of culture of science and how maybe it warms more to mechanistic approaches as opposed to organistic approaches?
Well, I'm not a sociologist of science, and that's not just an excuse, because I think that's a serious question which requires a serious answer, and I think it's a question which requires some empirical research. I mean, my sense is, you're right, my sense is that the culture of science is mechanistic at a certain sort of level, unless you go to, Dartington Hall or some of these places which set out deliberately to work on, Forest Row, where all the Steiner people are, they're doing all this biodynamic agriculture and that sort of thing, Emerson College, and they are very deliberately trying to inculcate a more organicist view. So there are places which are trying to do that, but they're trying to do it, and they're obviously very much minority places. So the answer to your question is, yes, I'm almost certain you're right. But it doesn't, it's one of those things, isn't it? Scientists are, what shall I say, two-faced hypocrites or something like that. They'll tell you one thing, and then they'll do another. I wrote a book on progress, and I realized pretty quickly, when I was interviewing biologists, I'd ask them about progress, and they'd be looking at my tape recorder, and they'd say, no, I don't believe in it. And then half an hour later, when they were feeling relaxed, they'd say, well, frankly, Mike, don't tell anybody tape recorders going, and I'd sit and they'd say, you know, I really think there's a lot to be said about progress. And my feeling is that there's a lot more empathy, as you pointed out. They probably don't want to call it that. Because if you call it that, you know that you're wet or you're a late developer. But my suspicion is, you know, there's more than one supervisor said to a student, you know, this is pretty good, but, you know, you really need to pull back and look at the big picture. But they wouldn't, and as I say, I don't. They wouldn't think of this as being anti-mechanistic in any sense at all. I mean, if you look, the fascinating one, let me finish on this point, is the extended phenotype by Richard Dawkins. My God, it's the most holistic book I've ever come across. I mean, here's a guy who's saying something that a bird does in Africa can have effect on parasites in Scotland by the end of the year. Now, if that isn't, at some level, trying to link everything together in one integrated whole, I don't know. And yet, this is the book where the guy's taking off after Gaia and thinks that what he's doing is writing a sort of a manifesto against organicism. Well, meanwhile, his own examples are things I'd say, hallelujah, you know, God really does exist. Well, I wouldn't quite say that.
Can I just say first, I mean, you know, I always love listening to you. Your talks, they're actually very informative. They're extremely entertaining. And I always like...
I'm not sure from a headmaster that that's a word of praise.
Absolutely. What I also like, I feel comfortable saying, is that nearly always, about 2/3 of the way through, I find myself thinking, why am I listening to this? And then by the end of it, it has all come together.
That explains why you slept for the first half.